as well. So next thing we're going to talk about is protein synthesis. So this is DNA, and DNA serves as the master blueprint for protein synthesis. We said DNA is made up of these nucleotides, and there are different letters, A, T, C, and G, and they have the information that we need to produce proteins, and we're going to talk about how that happens. Genes, we don't know what a gene is. You've probably heard that term. Genes are segments of DNA carrying information needed to produce specific proteins. So you might hear about an eye color gene. Really, that gene is producing specific proteins that are going to influence eye color. This is an example of how DNA codes for proteins. Remember, we said proteins are made up of amino acids. So for example, if the code on the DNA is AAA, there's two strands to DNA. So one strand will be AAA, the other strand will be TTT. mRNA will be produced, and it will be UUU. And the tRNA that is going to bind to this is going to be AAA, and that codes for phenylalanine, which is one of the amino acids. And you don't have to memorize this chart or anything. I just wanted to give you an example of how it works. So in this process, information goes from DNA to RNA to amino acid, which becomes a protein. A polypeptide is a protein. So again, this process right here, this is known as the central dogma of biology. So it's pretty important. It's why life exists. We go from DNA to RNA to amino acid. So the first thing that has to happen is called transcription.
The innermost workings of how a simple code is turned into flesh and blood. This is what Francis Crick called the central dogma of modern biology. How DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in the body. When the RNA copy is complete, it snakes away from the nucleus into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another number.
Okay, I love that movie. It's the best explanation for transcription I've seen. So what transcription is the transfer of information from DNA to RNA. And DNA and RNA are the same, except RNA uses uracil instead of thymine, U instead of T. So the messenger RNA, which is what we saw being produced, that was the yellow thing being produced. And again, it's being produced so fast, and that's happening right now in all of your cells. It's pretty amazing. So that the blue molecule we saw is called RNA polymerase. So RNA polymerase does a few things. First, it unwinds the DNA template. Then it adds complementary bases on the DNA template. So here it is. Here's RNA polymerase, and it's kind of unzipped this. And then it's going to start to add RNA. That's the complementary bases, and it joins these together until you've got this long strand. Then it's going to receive a stop codon. When it reads the stop codon, it's going to stop. That protein's complete. And then that mRNA will get edited before it leaves the nucleus. So these parts that aren't necessary, they will be what we call spliced out. They probably have some regulatory function that we don't understand yet. So that's transcription. That's how we go from DNA to RNA. Then the next process we're going to talk about is translation. And that happens at the ribosome.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
Okay, so that is the process of translation. So in translation, transfer RNA, or tRNA, binds to amino acids. So that's what this molecule here is. It would be a transfer RNA. It binds to amino acids and brings them to the mRNA at the ribosome. Here's the ribosome. So the mRNA is produced in the nucleus, and that process is called transcription. Then it goes out to a ribosome for translation. So the tRNA brings in an amino acid, and they're joined together, as you saw in that movie. The codons, this right here, 3 nucleotides is called a codon. That pairs with an anticodon. And that's how we ensure we get the correct amino acid there. So we make the right protein. And the amino acids are joined together by peptide bonds. And they form these polypeptide chains. And then there's a termination signal, and it stops, and our polypeptide is done. And that is the process of translation. And there's the whole process. So now I have some review questions. The process of making RNA from DNA is called, and hopefully you got transcription. The process of making proteins from RNA is called, and hopefully you pick translation. And which molecule takes information from the nucleus to the cytosol? And the correct answer would be mRNA. The DNA all has to stay in the nucleus, but we need to get the message out to the ribosomes, and mRNA is the molecule that does that. Which molecule binds to an amino acid and has an RNA anticodon on the other side? So this is an RNA molecule that binds to amino acids, and it's the tRNA that does that. Okay, so hopefully you already did that, labeling the cell, labeling the organelles from that cell picture and giving their function. You don't need to turn that in, but that's going to help you study. Next thing we're going to talk about is how things enter and leave the cell, and we're going to start with a movie that's found on Mastering A&P. So I'm going to log into Mastering A&P, go to the study area, and over here on the left I'm going to choose A&P Flicks, and click A&P. And membrane transport is what we're going to watch.
In order for us to use the molecules and energy we get from food, we must transport them across cell membranes. Transport of nutrients is essential for all cells, but we'll use cells in the small intestine to teach the principles of membrane transport. Let's zoom into the surface of an intestinal cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can move freely across the lipid bilayer. Notice that the lipid molecules move down their concentration gradient from where they are more concentrated to where they are less concentrated. This is an example of simple diffusion. Diffusion is a form of passive transport. It does not require energy from the cell, but it does depend on the concentration gradient, size and charge of the molecule or ion. However, most molecules cannot cross the lipid bilayer. Instead, molecules like the sugar fructose move into intestinal cells by facilitated diffusion, moving down their concentration gradient and through transport proteins. It also differs because it acts only on specific molecules and the transport proteins can become saturated, reducing the amount of diffusion. However, facilitated diffusion doesn't require energy from the cell, so, like simple diffusion, it's a form of passive transport. The majority of water molecules cross the plasma membrane by facilitated diffusion, also called osmosis, through specific protein channels called aquaporins. Some ions, such as sodium and potassium, require transport against their concentration gradients. The sodium-potassium pump actively transports these ions from where they are less concentrated to where they are more concentrated. This process requires energy from the cell and is known as active transport. Energy from ATP is used to move 3 sodium ions, shown in red, out of the cell, and 2 potassium ions, shown in blue, into the cell. Because the pump uses ATP directly, this type of transport is called primary active transport. Another type of active transport, secondary active transport, also moves chemicals against gradients. It doesn't use ATP as a direct source of energy. Instead, it uses the downhill gradient from one ion or molecule to drive the uphill movement of another ion or molecule. For example, sodium and glucose move into the cell by secondary active transport in a process called cotransport or symport, because both substances are moving in the same direction. Sodium ions move down their concentration gradient created by the sodium-potassium pump, but because glucose concentrations inside the cell can quickly exceed those outside the cell, glucose moves against its concentration gradient. The cell moves larger materials, such as proteins, from the intracellular fluid to the extracellular fluid, using vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. Cells use all these processes, passive and active transport, exocytosis and endocytosis, to move materials across their membranes.
Okay, and you can go back to Mastering A&P and watch that again if you would like. You can watch it as many times as you want. It's a good overview of the process. So again, our plasma membranes are selectively permeable. So things like, well, this is not our cell membrane, but this is an example of a selectively permeable membrane. This one lets ions, water, and carbohydrates through, but not lipids and proteins. So the way things can get through our cell, there's basically passive mechanisms and active mechanisms. Active mechanisms require energy, or passive mechanisms do not require energy. And our cell likes the passive processes because, I don't want to say they're lazy, but they're efficient. So a lot of things cross the cell membrane just by diffusion. And we're going to watch this.
Water vapor spreads
that
out
talks
into
about
the
diffusion.
atmosphere. It's carried by the wind, but it can also spread out of its own accord by a process known as diffusion. Invisible gases behave in the same way. The particles in a gas move randomly in all directions. This helps explain why a gas diffuses from one place to another. You can see diffusion happening on a small scale in the lab using bromine, an orange-coloured gas. Place a jar of air on top of the bromine, and it slowly starts to spread. It's moving up the tube against gravity. Over time, the two gases mix. After half an hour, both containers have a uniform colour. It takes time because the air and the bromine molecules are colliding with themselves and each other. So what would happen if there was no air for the bromine to bump into?
Okay, that's a brief overview of diffusion. If you've ever made Kool-Aid, you've probably seen diffusion happen. Molecules don't like each other, so they spread out. That's diffusion. Things spreading out, and we say things go down their concentration gradient, or they go from a high concentration to a low concentration. Simple diffusion. So this would be simple diffusion right here. Nonpolar and lipid-soluble substances can diffuse directly through the bilayer. And then there's a special type of diffuse. So simple diffusion, things can go right through. There's a thing similar to diffusion that we have another movie about.
This membrane separates pure water on the left from a solution of sucrose and water on the right. The membrane is semi-permeable. Water can cross it, but sucrose is too large for the membrane's pores. Water molecules will move from a solution with a lower concentration of solutes to one with a higher concentration of solutes. A solution with fewer solutes has more water molecules that are free to move about and follow their concentration gradient across a membrane. Thus, there will be net movement of water across this membrane from left to right. This diffusion of water across a membrane is called osmosis. Osmosis is of great importance in the water balance of all cells.
Okay, so osmosis is basically the diffusion of water, and it occurs when the concentration of water is different on opposite sides of a membrane. So osmosis, or diffusion of water, occurs when the concentration of water is different on opposite sides of a membrane. Now this shows what would happen to a red blood cell in different solutions. This first solution is called an isotonic solution. A solution with the same concentration, so it's not going to cause net movement of water in or out of the cell. That's known as an isotonic solution. A solution with a lower concentration, so that would cause net movement of water into the cell, is known as hypotonic. And the problem with that is water would enter the cell and it would swell up and eventually it would burst. And if it bursts, it can't do its job anymore. Then a solution with a higher concentration would cause net movement of water out of the cell, and that would cause the cell to shrivel up. This is called a hypertonic solution. And if your cells shrivel up, they can't do their job very well. So those are some terms to know. Isotonic, hypotonic, and hypertonic. You'll see them later on. This is very important when we give, for example, IVs. If you've ever gotten an IV, what they give you, it's not water, it's a saline solution. About 0.9% saline, which is isotonic to our body cells. That way they won't cause your cells to burst when they give you IV fluids. Now if you're dehydrated, they might give you a hypotonic solution, but they would be careful about it. They wouldn't inject pure water into your veins. Alright, so back to how things get through a cell. How things get through a cell membrane. This is called facilitated diffusion. So glucose is too big to go through the plasma membrane. So the cell makes carrier proteins, and they allow glucose to enter. It's still going down its concentration gradient, so it doesn't require energy from the cell. So the cell likes this. Facilitated diffusion uses integral transmembrane proteins, and they usually have a binding site for a sugar or an amino acid. Then the next type of transport is called active transport. Active transport can move things against their concentration gradient. It uses ATP for that energy, and it requires carrier proteins. There are two types of active transport. Primary active transport, which is what you see here. This protein uses ATP to pump things against their concentration gradient. So this one here is pumping sodium out and potassium into the cell. And it uses ATP to do that. So that would be primary active transport. We're directly using ATP. That movie also showed secondary active transport with glucose and sodium. So by pumping that sodium out, this protein here set up a concentration gradient for sodium. And now the cell uses that to bring glucose into the cell. So secondary active transport uses an exchange pump to indirectly drive the transport of other solutes. So this sodium gradient that's set up by this pump using the energy is used to bring glucose into the cell, and the cell needs the glucose. Alright, so which of these is not a passive process? And the correct answer would be active transport, because active transport requires energy. And diffusion of water across a membrane is called, and the correct answer is osmosis.
Cells also use energy to transfer materials in bulk, but this time by forming membranous sacs that hold their contents under wraps. In a process called endocytosis, part of the surface membrane encloses the material, forming a sac which brings the contents into the cell's interior. In the opposite process, called exocytosis, the sac moves through the cytoplasm to the membrane, fuses with it, then releases the contents.
Okay, so that movie gives an overview of exocytosis and endocytosis, and you can see how the membranes of the vesicles and the membrane of the plasma membrane just kind of fuse together. So this figure here shows what's called receptor-mediated endocytosis. Sometimes our cells need a certain nutrient. One example of this is cholesterol, and your cell will put receptors for cholesterol out into the plasma membrane. And then they'll bind to the ligand, whatever it is, and then it will pinch off this vesicle and bring that into the cell. And then it can be fused with a lysosome to release those. And then the cell can use these nutrients, and then it will take those receptors and put them back into the membrane and bring more of this substance into the cell. So that's receptor-mediated endocytosis, and your cells can do that if they need something, such as cholesterol. This is pinocytosis, sometimes called cell drinking. When we get these tiny vesicles pinching off the cell membrane and bringing something through the cell, bringing something into the cell. Phagocytosis is sometimes called cell eating. It's bringing a large thing into the cell. So in this case, we're bringing a bacterium into the cell. And we're going to destroy it with a lysosome and then recycle the nutrients and get rid of the stuff we don't want. And then exocytosis, I have a cool movie of exocytosis happening in a real cell. So watch down there. You'll see things leaving this cell. So way down there. So if you look right in there, you'll see things leaving the cell. If you're a single cell, that's how you get rid of wastes, by exocytosis. So this really does happen. We're not just making it up. Okay, so which of the following describes materials leaving a cell in a vesicle? And we just watched a movie of that, so hopefully you remember that is exocytosis. Which of the following describes large particles being brought into a cell in a vesicle? And a large particle or cell eating is called phagocytosis. Alright, next thing we're going to talk about is the cell life cycle. So this is the life cycle of a cell. You can see it spends most of its time in interphase, and division of the nucleus is called mitosis, and then division of the cytoplasm is called cytokinesis. So division of the nucleus is mitosis, division of the cytoplasm is cytokinesis. Then there are some different phases. There's the G phases, which are for growth. And then during this S phase, the DNA is replicated or copied. So we're going to talk about that next, how this DNA is replicated. And
During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. Copying occurs at a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step in DNA replication is the separation of the two strands by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems. The separated strands are called 3' and 5', distinguished by the direction in which the component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite orientation and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding proteins, and in some systems, multiple Okazaki fragments may be present.
okay, so that movie is a good overview of DNA replication. There's also a movie again from the Master AMP site, AMP Flicks. DNA replication. This is another good animation.
Prior to cell division, the genetic information which is stored in chromosomes must be copied in a process called DNA replication, and the duplicated DNA separated evenly between two cells. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in the large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the two strands of DNA separate and then serve as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming two replication forks. In humans, numerous replication bubbles are formed. Let's zoom in on one of them. Some of the proteins that work together at the replication fork are shown here. Here, DNA helicase unwinds the two strands, and DNA polymerases type 3 build new strands of DNA. A critical component of the DNA polymerase III protein is the DNA clamp protein, which prevents the polymerase from dissociating from the template strand. Thus, the process of DNA replication is called semi-conservative. Each new double helix contains one original DNA strand and one new DNA strand. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously and forms from its 5' end to its 3' end. The orientation of the other new strand, the lagging strand, is opposite to the working orientation of DNA polymerase III, and thus the lagging strand must be built in fragments, called Okazaki fragments, in the 5' to 3' direction using multiple primers. First, let's focus on the leading strand. DNA polymerase III builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands. Because of the opposite orientation, replication of the lagging strand is more complicated. To initiate synthesis, an enzyme called primase reads the DNA strand and produces a short segment of RNA, which serves as a primer. A DNA clamp protein surrounds the DNA and attaches to DNA polymerase III, which builds the rest of the new fragment of DNA until it reaches the previously synthesized Okazaki fragment. When the fragment is finished, the polymerase releases from the Okazaki fragment. How are Okazaki fragments of the lagging strand joined together? A different DNA polymerase I, removes RNA and replaces it with DNA. However, it cannot finish connecting the Okazaki fragments. An enzyme called DNA ligase joins the Okazaki fragments together. Growth of the leading and lagging strands continues at both replication forks until there are two identical, double-stranded DNA molecules. Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells.
Okay, so that's how DNA replication happens. And that's enough. That's as much as we need to know for this course. So again, you can go watch that again if you'd like. So briefly, each DNA strand serves as a template for building a new strand. The DNA polymerase binds and adds new DNA nucleotides to the template strand. And replication is just copying of DNA. Don't worry about the Okazaki fragments. And here's our final product. We've got two new DNA molecules that are exact copies of each other. Next topic is mitosis, which we said earlier is division of the nucleus. And we have another movie for this. And we're going to come back to this picture after we watch the movie. So mitosis. And you guys can come back and watch these as many times as you want.
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
Okay, so this dog, he peed on the mat, and if he would talk, he would say, "I pee on the mat," see? And that's how I remember the phases. There's interphase, prophase, metaphase, anaphase, and telophase, and then C for cytokinesis. And so you should know those phases and what's happening in each phase. Alright, last thing we're going to talk about, we're not going to go through all the phases right now. The movie did that for us. So, what must happen before a cell can begin mitosis? And the correct answer is that the chromosomes have to be duplicated. The centrosomes move away from each other and the nuclear envelope breaks up, during which phase of mitosis? And the correct answer is prophase. If you're ever not sure, just guess prophase, because that's when the most stuff is happening. The chromosomes line up in the center of the cell, during which phase of mitosis? And the correct answer is metaphase. The sister chromatids separate and begin moving toward opposite poles of the cell, during which phase of mitosis? And the correct answer is anaphase. Chromosomes arrive at the poles and nuclear envelopes form during which phase of mitosis? And that would be the last phase, which is telophase. And at the end of the mitotic phase, the cytoplasm divides in a process called cytokinesis, is the division of the cytoplasm. Alright, then we need to talk about cancer. Cancer is an illness that disrupts normal rates of cell division, and cancer causes all kinds of problems. But this is really what starts all these problems. You get one abnormal cell that starts to divide, and it's dividing rapidly when it shouldn't be, and then we've got all these cells, and they're all dividing rapidly, and it grows and grows and grows until we have a tumor. It's characterized by permanent DNA damage, DNA sequence damage, or changes, and we call these mutations. It's most common in tissues with actively dividing cells, such as the skin or the intestinal lining. Skin cancer is common, colon cancer is common. Those tissues have pretty rapidly dividing cells anyway, so we think it's easier for those cells to get out of control. These abnormal cells, they compete with normal cells for resources and cause problems. So first, this would be an example of a neoplasm or massive cells, a tumor. This would be a benign tumor. A benign tumor is going to remain in its original tissue. It's not going to spread. That would be the least dangerous type of cancer. A tumor like this one that is attracting blood vessels to itself and then sending out cancer cells into the circulation, this would be a malignant tumor. These grow faster because they bring a blood supply into the area. So one approach to treating cancer is try to stop the formation of new blood vessels. And the processes, when invasion happens, that's when cells migrate into the blood circulation, and then they'll escape. And when they form a secondary tumor, that is metastasis. And we'll talk more about cancer later. Here's a person with a malignant tumor on their thyroid gland. So an illness that disrupts the normal rate of cell division is called, the correct answer is cancer. A tumor that increases blood supply to an area and spreads to surrounding areas is called malignant. And I believe that's all the questions I have. So email me if there's any questions from this movie or suggestions. And I will see you in the next chapter.